Alright, so let's give this a shot. We're going to talk about incomplete dominance today. So, I know this is weird because I'm not really in the classroom with you, but we're going to give this a shot. I'm going to try to record a lecture. And doing that, I've got the PowerPoint out, the same PowerPoint we have in class. I adjusted it a little bit so I could talk to you about this one little concept you're going to do today. So you should have a notebook out, and you should be listening. Okay, pay attention. You know I can't draw, so that's the best I'm going to do. And the nice part about the video is if you do get stuck on something, or if I'm going too fast, you can raise your hand and the substitute can pause it, rewind it. You can ask questions, see if someone else in the room knows the answer. If you're still confused about Punnett squares, hopefully you can straighten some of that out today. And the only other thing I would say is during the course of this little lecture here, it's going to be less than 10 minutes, I think. If you do get stuck, if you have a question, you really, really don't know where to go with it, write it down. And when I see you in next class, you can ask me. Make sure you get your questions answered. I picked incomplete dominance because it is one of the easier exceptions to the rule. You'll see in a minute. It does not follow dominant and recessive traits the way all of the basic Punnett squares you've done up to this point follow. And I'll show you one good example of that right now. So you should have your notebooks out. You should be ready to write. And again, if I go too fast, just you can pause it. It's no big deal. Okay. So here we go. We're going to use one real example of incomplete dominance. Down in the bottom right hand corner, those really pretty flowers, they kind of stack up in little columns along the stem of the plant. And they have a gene that is incompletely dominant for flower color. So it makes it nice because it's something we can really see. And the name of that plant is the snapdragon. So florists use them a lot. In bouquets, you see them all the time. And the first genotype shouldn't surprise you. If you have homozygous red, remember you have 2 alleles for every gene. So if you have red, flower ends up being red. No surprise. Same thing if you're homozygous white. If you have 2 white alleles, you end up being a white flower. But in this case, in incomplete dominance, instead of one of the colors winning, if they're mixed together, if you have a red allele and a white allele, the flower ends up being pink. So the flowers and all that stalk would be pink. I believe those are the red ones right there. I think the pink's a little lighter than that. So when you do these genetics problems, you've got to change the letters you use because an uppercase letter like a big R and a lowercase letter like a little r, they won't work anymore because you don't have a true dominant, a true recessive. So we just change how we write, what letters we use. And you're going to see that in a second. So we'll go to the next slide here. And if you write out your genotypes for incomplete dominance, this is what they look like. And I had them slam into the PowerPoint because these are really important that you understand the difference. Now instead of using the same letter, as I just said a minute ago, now we use 2 different letters. We use R for red and W for white. So when you write out your genotypes, you end up with something a little bit different. If you have RR, obviously you're red. It's the only 2 alleles you have are both red. It's the only thing the flower could be. Same thing with WW. If all you have are 2 white alleles, the flowers are W. But in order to mix them, if you get an dominant and recessive, if you remember, we would call this a heterozygote. We don't really call this that. We don't really call it a heterozygote anymore. Instead, we just say if you're a mix of the 2 alleles, an R and a W, you make pink. And you can still set up a Punnett square. You run the Punnett square exactly the same. You just have to use this genotype key instead of using an uppercase and a lowercase letter, using 2 separate letters. So setting up a key like this for incomplete dominance is probably a good idea, and it's probably the way to go. So let's see what happens. You get a genetics problem going on. So here's our first problem. We're going to cross a red snapdragon with a white. So step 1 would be to choose letters. Well, they're already over there on the left side of the screen. So we're using R for red, W for white. It's incomplete dominance, so R is red, W is white, R, W together is pink. So step 2 would be to assign upper and lower case. We're skipping that because you don't do that. Step 3, figure out what the parents are. Well, we have a red parent and a white. And again, if you look at the key on the left, it's already done for you. We're crossing R with WW. Now you set this up, you run the square exactly the same as you would with a dominant and recessive. So I'm going to put my one parent's alleles above the boxes. I'm going to put my other parent's alleles on the sides. So if you look, they went just like they did before. I didn't put arrows in for the W's because it gets a little crowded on the PowerPoint. I took them off. And now we just got to put them in. So take a minute. What I would do right now is I would actually pause this, let everyone fill it in, and then look at the answer in a second. So go ahead and pause it. Alright, so hopefully you paused it. And for those of you that are really comfortable with Punnett squares, you probably figured this out really, really quick. All you do, you're going to pull the top R is going to come down into the lower two boxes. The W's are going to shift over to the right. So here they come. So your first box is going to be RW. Your second box, same thing, RW. Third box, RW. And fourth box, RW. So what do you have? What you're going to have is if you cross a red snapdragon with a white snapdragon, all of your offspring are going to be, guess what? They're all pink. They're all RW, and if you look at the key to the left, they are all pink. There's no way around it. That's the way it's going to be, because this time the traits blend. It would be like tall and short made medium, if you guys remember on your Alien Encounter Lab, first trait. Okay, if you were a head and a tail, so you were big, little for height, you ended up being medium. You ended up being a blend of the two traits. It's not a true dominant or recessive. No one's winning here. Instead, they are blending together to create another phenotype. Right, so, and here's a little image. If you took red snapdragons and you cross them with white, you end up with pink. So let's do another problem. All right, this one we're going to change it up just a little bit. We're still using our snapdragons because you still have the key right there in your notes. It's easy to see and follow. We're going to cross two pinks. So we're going to use still R and W. Why change it? I already have a key. The first thing we have to do is figure out what our parents are going to be. And I just copied the key from the other slide, so in case you're not writing this down, you can still see it. So our two parents are RW with RW. If you look at the key, RW is pink. So here's your square. Set your square up. All right, and what I would like you to do as a class, if you could, pause it now, run the square, talk about the answer you would get. What would you get out of this? What would the offspring be? And then replay it, and I'll go over it. So go ahead and pause it now. All right. So here's our first parent. Here's our second. And again, if you fill in, your first offspring is RR, which is red. The second one, the R comes in from the left, the W drops down, you get pink. You get the same situation in the third box, and the last box, you get white. So now, again, remember in genetics, you always have to answer the question. I don't have a question up here, but you can kind of assume the question would be, how many of the offspring would be expected to be pink? Or what would be the ratio of red to pink to white? So I just went ahead and put that over here. You get a 1 to 2 to 1 ratio. You get 1 red for 2 pinks to 1 white. So now you have a second way, other than dominant and recessive, to look at really basic genetic problems. And there are several of these in your packet. There are some, there's an entire page on bikini bottom genetics that deal with incomplete dominance. I'd like you to work on those. And the Oompa Loompas from number 7 on all deal with this idea of incomplete dominance. And I'd like you to work on those. You can work together in a group of 1 or 2. I don't really expect you to turn this in. Again, if you're very comfortable with Punnett squares, you should go through this fairly quickly. If you're uncomfortable with Punnett squares, take your time. I would make the key, like the RR is red, WW is white, RW is pink, before you do anything. And hopefully that'll help you out. So good luck with this. If you have any questions, write them down. I'll talk to you when we meet again. And good luck.